Now I have the honor to invite the second speaker. We will have plenty of time, Professor Nick, for a very stimulating discussion later during the open forum.
Good morning, ladies and gentlemen, President Pascual, former President Nemenzo, esteemed faculty, colleagues, students, guests, and friends. In this talk, I would like to introduce to you some basic knowledge of chemistry, biochemistry, and evolutionary biology that could provide insights for social and institutional change. Hopefully, this can help spark new interdisciplinary research among us. So these are the objectives, and this is the flow of my talk. Physico-chemical principles that apply to biomolecular structures and functions that underlie complex biological systems and evolutionary biology. Then let's take a look at similarities with human life and social and institutional systems. Some of you are familiar with what is in this slide and the coming slides. What I will do is to simplify it for those in the audience who belong to other disciplines. In the whole universe, we know that the total energy is fixed. This is a first law of thermodynamics. And that entropy, which measures disorder, is increasing in the universe, the second law of thermodynamics. Now, let's look at the energetics or thermodynamics of change. This free energy change or delta G equation tells us if a chemical reaction can occur spontaneously. That is, if it moves towards equilibrium. And we know that equilibrium is a state of maximum entropy and lowest free energy. If delta G is negative, the change will occur. Delta G has two terms, delta H, or the change in enthalpy or total energy, which contributes positively to delta G, and delta S, the change in energy associated with randomness, disorder, or entropy at a given temperature, which opposes delta H. Intuitively, we expect a decrease in total energy or a negative delta H, and an increase in entropy, positive delta S, to be spontaneous. But within the universe, such as in life, energy can come in or out and is not fixed. Life is an open system, not an isolated system like the whole universe. And also in life, entropy can decrease or increase, just like in these chemical reactions. So these are chemical reactions, and let's take a look at reaction B, where the decrease in enthalpy is so large and offsets the decrease in entropy. So reaction B. So why am I telling you this? Because we can relate energy and entropy to change or resistance to change in human life and social systems. In living systems, many biomolecular structures are highly ordered or have a low entropy, such as a protein structure. So low entropy structures are here, aside from proteins, a diamond, very structured, a Shakespearean sonnet, a bank manager's desk would be a low entropy. Forming that protein must have entailed a decrease in entropy. Now, think of our metabolism. We are able to build biomolecules with highly ordered structures such as proteins and DNA, and for this we need energy, which we get from breaking down the food we eat. This is possible, again, because life is an open system. Proteins and DNA fold and refold, self-organize to create a hierarchy of ordered structures, and then form assemblies of or complexes of these structures, which make up cells, tissues, organs, and organisms. Proteins have a 3D structure with specific binding sites, and a protein has the ability to move its parts or change its conformation. Take the case of hemoglobin, which takes up oxygen in our lungs, gets transported through the arteries, and gives off oxygen in our tissues. In this cartoon, So we call these cartoons in biochemistry. Hemoglobin has 4 highly folded units that interact closely with one another. So you have the red, the blue, and 2 more at the background. A simpler representation of the 4 units would be these 4 boxes. When one hemoglobin unit picks up an oxygen in the lungs, it changes its structure and influences the 2nd, the 3rd, and the 4th units to each pick up more oxygen. This behavior is known as cooperativity or synergy. The reverse process occurs when one hemoglobin unit releases oxygen in the tissues. The 3 other units cooperate and also give up their oxygen. Hemoglobin shifts between the R state, the oxygen-rich state, so here you have oxygen represented by the red dots, and the T state, or the oxygen-poor state. It's also represented by this graph, which is reflective of a sigmoidal switching behavior between the strong oxygen binding in the lungs and the weak oxygen binding in the tissues. Now back to the cartoon. Look at the change in position of the 4 units relative to the vertical line in oxygen-rich, and deoxyhemoglobin. So it involved some conformational change. To summarize, hemoglobin demonstrates the specific binding properties of proteins. Proteins create 3D spaces or niches. And proteins, such as enzymes, also create time. Enzymes can catalyze chemical reactions 1,000 to 1,000,000 times faster than an uncatalyzed one and at low body temperature. This saves the cell a lot of time. And like a molecular machine, the enzyme goes through a catalytic cycle and keeps churning out products from the reactants. Now think about how we metabolize glucose as a source of energy. It first needs to be activated by a phosphate before it is broken down. The enzyme hexokinase brings glucose and ATP, which is the source of phosphate, together in its active site to catalyze a chemical reaction. Look at the conformational change of hexokinase when glucose binds it. So there's a real change to embrace the glucose. This results in a lower activation energy of reaction, which translates to much faster kinetics. So while energetics can predict whether a reaction occurs spontaneously or not, it does not predict the reaction rate. And for that, you need to look at the path of the reaction. So I'd like to echo what Professor Nick said, that in complexity, we have path dependence. So through complex dynamical systems and structures, nature has found bioenergetic and kinetic ways to make life processes very specific, very efficient, very productive. A cell is made up of subcellular organelles and spaces containing assemblies of functionally linked biomolecules. And because of this, the cell is capable of self-perpetuation and self-renewal. Complex organization and interactions of cellular components form signaling pathways, resulting in complex dynamical behavior, and the cell is generally robust and resilient when it suffers stress or damage. A cell is likened to an electrical circuit with certain proteins behaving like sensors, transducers, adapters, amplifiers, resistors, and rheostats. However, in some instances, it can suffer a catastrophe from which it cannot recover because of damage of certain key genes encoding key proteins. To the right is your normal cell, like a circuit, showing key proteins and factors like the p53 protein, which is a DNA damage sensor. p53 is a tumor suppressor protein that is known as the guardian angel of the genome. p53 is our frontline defense leader. And when it senses DNA damage, it stops the cell from dividing, tries to repair the damage, and if damage is beyond repair, the p53 orders and programs the cell to die. To the left, you will see the cell with many more proteins shown in the circuit. Those in red are proteins that have been found mutated in various kinds of cancers. And among these, mutant p53 is the one found in the most number and kinds of cancers. Mutant p53 is no longer able to behave properly. It's not able to retain DNA, repair DNA, and so progressively, more proteins are mutated in a cell and it turns cancerous. So far, we've talked about principles underlying molecules and cells that apply to human life and social systems, not just as metaphors, as Professor Nick mentioned, but in real terms. I talked about open systems that allow movement of energy and resources, stability versus disorder, which applies to societies, cooperation versus competition among individuals, creation of niches or priorities, productivity rates and progressive change, feedback looping and control, signal amplification, organized structures that are robust and resilient, assaults on key components leading to catastrophes, and more. Now let's take a look at evolution, which is biological change over a long period. Darwin's and Wallace's evolution was based on morphology, the empirical observation of form and growth and development of organisms that they found in an isolated environment, like the finches in the Galapagos. Natural selection for the best variants with the best form and function, which led to speciation, was based on competition in the environment. The variations were gradual, slight, and successive. In the case of Darwin's finches, the radiation into species with different bills was based on adaptation to their diet. Now, those who question evolution raise the following issues: gradual change versus saltation, simple to complex, small to large structures, and the question of irreducible complexity, which would require parallel or simultaneous evolution of parts that come together to make a whole, for example, the complex human eye, which would thus require intelligent design. Okay. Today, we understand evolution in molecular terms and not just in morphological terms, and so we can explain these issues better. Take the case of the simplest, tiniest organism, a single-cell bacterium, Salmonella typhimurium. It has a hook-type hollow flagellum sitting on a base of many, many proteins assembled together, which it uses for locomotion. This motor is yet unmatched by human engineering. It's very complex, but it is tiny, and it is evolutionarily old. Okay? It's one of the earliest forms. This has been used as an argument against evolution by creationists, that complex structures cannot evolve from simple structures, that it is irreducibly complex and need all of the protein components to function. However, it has been shown that some of the proteins can be deleted without complete loss of function. And it is generally accepted that bacterial flagella evolved from much simpler secretory systems. So this is telling you that there is complexity even in the molecular world, but the complexity can be broken down into prototype parts or components. And the basis for all of this would be molecular biology. So we now have here the central dogma, which has been modified for general and special applications. And we have information flowing through biological systems. And while I had shown examples of biomolecules that result in amplification effects, here we have multiplicative effects. So through genetics, you are able to get more of the effects retained through evolution and development.